thanks, Richard, for the invite. The last time I gave a talk here was in 1997. I promise the slides are a little bit updated. So this is going to be a blend of mathematics and neuroscience and engineering, and there'll be a little bit of intro about imaging and how it's used to map brain disease, and then we're going to go deeper into the mathematics and the engineering aspects of some of these problems. So the ENIGMA Consortium is a network of people around the world that collect brain images. And it's very unusual to club together to do a very large brain imaging study. But one of the incentives that made it interesting for people to pull data was to try and discover links between our genetic makeup, our DNA, and brain function and connectivity, and some of the cognitive functions that we have. So I'll talk generally about that, but I'll give you a little bit of an intro. So just before we start, we're very glad to be here. You guys have been very, very warm and welcoming to us. Our lab is at the ISI at Marina del Rey. There are 4 junior faculty who are here today. Thanks for coming. And we do large-scale studies using imaging of Alzheimer's, depression, schizophrenia, bipolar. There's a lot of work on big algorithm development, so there's a team that does image analysis for scouring and collecting information from images. And our center is part of a new Institute for Neuroimaging and Informatics at USC that's at the Health Sciences campus. And so there'll be a new 7 Tesla scanner installed at the end of summer. There's a lot of scope for doing innovative high-field imaging projects as soon as that's underway. This is also the hub for other large-scale databases. And so people interested in informatics and mining databases for patterns and information, there are some large-scale projects that are handled out of that. All of this is now at USC. So what is ENIGMA? Well, ENIGMA was a project in the Second World War for breaking the Nazi code, but we're not doing that. It's the largest brain imaging study in the world. So one of the papers in Nature Genetics, beginning last year, reported MRI data from 21,000 subjects. It's now larger. And it's a worldwide partnership pulling data from the US, Europe, Australia, Asia, and Africa. And some of the goals are to discover genetic variants in our DNA. We all differ in our DNA that affect the function of the brain, or that affect our risk for disease. We're also interested, regardless of genetics, in looking at global patterns in how diseases affect the brain. So if you're HIV positive, the virus gets into the brain, there are brain changes. Is that the same brain changes if you live in Africa versus the US versus Hawaii? So this is a global effort. And ENIGMA has working groups on different psychiatric disorders. So we're certainly not experts in these mental illnesses, but people who are leading them and putting together large-scale projects looking at that data. So to join ENIGMA, these are the sites. You need a brain MRI data set. It doesn't have to be from patients. It could be from healthy folks. And you can work with all these other people in trying to put together information from the scans on different topics. There's additional projects if you have genome-wide scans, GWAS, and I'll talk to you a little bit about what that is. So I'll give you some background on detecting disease effects with imaging. And I'll show you how that extends to detecting single gene effects on images, which is a little more unusual. We'll also talk about analyzing brain connectivity and doing statistics on the brain's networks. So that networks are an interesting topic. There's a lot of mathematical challenges. So we have a large engineering team. We're very keen to have engineering students join us. A lot of these signals are small. And so if you look for a gene effect on a large database of images, you need machine learning, sparse methods, ways to prioritize gigantic computations in images, adaptive methods to look at brain connectivity. I'll talk about those a little bit, a lot of concepts in mathematics. And also distributed computation. A lot of these sites don't share their data with us. And so there isn't a database here with a very large number of scans. A lot of the data is remotely in these different countries, and algorithms are sent out, and they're aggregated here. So there's a field called imaging genetics. Our center is called the Imaging Genetics Center. So we all differ in our genetic makeup, and you can pay $99 and send a saliva sample to 23andMe or a genotyping company, and they'll give you 1 million letters of your genetic code. They won't give you the full genome sequence, although maybe soon they'll be able to. And from that, you can see how you differ from the average representation of disease risk genes. And until a couple of weeks ago, they would give you all kinds of health-related advice, which they're now banned by the FDA from doing until they satisfy some of the compliance issues. Some of the questions are which common DNA variants affect disease risk, and many of you guys in genetics have been working on these questions. Another question is how do genetic variants affect the brain, and can we screen brain images to figure it out? And also, can we just pull data from around the world? I mean, a lot of the issues with brain imaging come from not having enough data sometimes to answer questions with satisfactory power. So here's a very simple example. So here's an MRI scan. Recording the surface, gray matter, white matter, fluid. You can make quantitative statements about whether this person's MRI scan deviates from the norm for people of that age and gender and so forth. So if you have Alzheimer's disease, amyloid plaques build up in the brain in this pattern. We can image amyloid, PET scans can image it, but on an MRI scan you'd see a pattern of gray matter loss. The thickness of the cortex would be thinner because the plaques cause neurons to die and there's a thinning of the cortex. And if you average data from lots and lots of Alzheimer's patients, you'd see a pattern where these parts of the temporal lobes, the memory systems, the hippocampus, would be smaller on average in a patient versus healthy matched controls. Now, you can take that a bit further. You can say, well, how about if I image patients with Alzheimer's disease who are just starting to get sick, or people that have had the disease for a couple of years? You can make a time-lapse movie where each frame of the movie is the statistical prediction for the thickness of the cortex for a patient who's had Alzheimer's disease for a certain amount of time. And so if you look at the early stages of Alzheimer's, the limbic system, the temporal lobe, these white colors are areas where patients differ from controls in the thickness of their gray matter. But as the disease progresses, there's this sort of flood into other regions, and there's associated cognitive changes. So this is practically very useful. If you ran a drug trial, let's say you had a blockbuster drug for schizophrenia, for example, you could give patients olanzapine, which is a new drug, or haloperidol, which is an older drug, and you could run a drug trial and say, well, does the newer drug save the brain from losing brain tissue? Is there any evidence that the brain tissue loss? Now, I got to say, in schizophrenia, it's controversial whether or not the brain differences are there. So there's a lot of interest in psychiatry in aggregating data from around the world to see if these patterns are consistent. But even before that's sorted out, they are already being used in drug trials as metrics of efficacy for different treatments. You can do epidemiology, so you can say, well, I'm interested in normal folks. Is there anything that predicts whether my brain structure will be more atrophied than normal or supernormal, so to speak? A good predictor is stress. You're very stressed, you have high cortisol levels, you lose gray matter in certain areas of the brain. You can study all kinds of things. You can say, well, what if people are overweight? If you are obese, there's a statistical linkage between your body mass and the amount of brain tissue in different regions. These are large studies, a cardiovascular health study, the ADNI study. So you say, well, okay, if you can find trends that relate things about us to images, what about drilling down into our DNA? So let's say we believe that your body mass index, your weight, is related to the amount of brain tissue. Geneticists discovered a gene called the FTO gene that obese people have a certain variant of it more commonly. One variant of the gene, if you average the weight of people with that gene, they weigh about 3 or 4 pounds more. You tend to crave about 200 more calories per day if you carry this variant of the gene. These are all averages. People are very different. So we knew that FTO was associated with body mass, and that body mass is associated with the brain. So it's an inescapable conclusion that carrying this gene is linked with something about the brain. So that's a gene-brain correlation that makes sense. Now, Meredith Bransky, who's here today, we were reading the discoveries of certain risk genes for Alzheimer's. So you often read in the newspaper, genomes mined for variants that are more commonly found in patients. Often we don't know what they do. So you can just mine a database of images and say, well, can you compare all the carriers of the risk gene that was discovered in the news, compare their scans with people that don't have it. And this is a DTI study that shows that carriers of a risk gene for Alzheimer's, even 50 years before you get the illness, have lower fiber integrity or factor fraction isotropy on a DTI scan. And you can keep doing this. So you can say, well, a schizophrenia risk gene, this is another one of Meredith's papers. People that carry this risk variant have lower fiber integrity on DTI. And you can keep going. So you can start to develop a polygenic test. So if you had your genome sequenced, if you pay $99 to have your genome code looked at, you can figure some kind of formula based on the genetic variants that you have that will be predictive of the types of differences in your brain relative to a population average. Now, how do you do the mathematics? Well, talk's going to get a little more mathematical. So each SNP in your genetic code isn't going to have much of an effect. So it's not something like a disease. I mean, the biggest effect it might have is about 1%. They're not huge effects. So what we do is we fit to the databases a statistical model that is a little bit like multiple regression. It's called LASSO. And it penalizes using a fantastic number of predictors. So if you're looking at a genetic code and you have 1 million letters available to you, you don't want a statistical model that has 1 million variables. You want to only have the ones that are useful. So you basically fit weights on your genetic code. This is a matrix of all the people in the study's genetic data. This is their imaging data. And you want to predict or minimize the difference between the genetic prediction and the imaging data, take the least squares deviation, and add a penalty, this is common in sparse methods or sparse regression, that prohibits you from fitting a model that's just fantastically complex. And you can predict about 6% of the variation in the white matter integrity of the brain just from these kinds of models. So could you find more genes that help or harm the brain? Now the typical way of doing this is that you find a large number of patients, maybe 10,000, 100,000 if you can, and you march along the genome looking for statistical overrepresentation of certain letters in that genetic code. I'm simplifying a little bit. There are other types of genetic variation that can be found. So there's a theory, and this is what motivated ENIGMA, that because genes affect the physical wiring of the brain, if you try to make direct connections between DNA and images, it might be faster. You might be better able to find genes that have a relationship to things that matter for the brain. And maybe some of the genes that affect the brain might affect disease risk. They could be good therapeutic targets. So here's ENIGMA. There was a large interest in pooling together brain imaging data sets from across the world where people's genetic code had also been evaluated. And that was the initial group of people involved. All of these folks are scanning both normal subjects and people with a variety of psychiatric illnesses, and they're analyzing their data, the MRI data and DTI data at each site. And there's collaborations with the Psychiatric Genomics Consortium that are looking at various different diseases. So before we go a little bit more into the mathematics, I'm going to tell you a little bit about the risk gene. So a risk gene is actually a variant in a gene where the spelling in your DNA is different. And there are catalogs of these, and you can look in the genetic code to see how these variants relate to images. So when I say a risk gene, I actually mean that the spelling of the DNA in a certain location is a little bit different. So here's the basic core of the mathematics. So let's say there was a gene that gave you a big blue head. Not an inflammatory example. You could look at people with big blue heads and say, well, tell me the letters that they have in their genetic code more than people with little blue heads. And there are 3 possible genotypes at each location. There's these 3. And you could run a regression that would allow you to make a statement about whether that letter in the genetic code had anything to do at all with the brain measure that you're looking at. Now, I want to introduce the idea of a Manhattan plot. This is a Manhattan plot. So this is the genome. And if you look at a certain spelling difference at a certain location, you could run a regression that relates that spelling difference to something you've measured in the brain. And you could say, well, if that genetic change or mutation or polymorphism is predictive of a higher value for something in your brain image, let's put the p-value for this regression on this plot. And this is the log-based 10 of the p-value. A lot of these things don't have much to do with the brain at all. If you beat a threshold of 10 to the minus 8, or basically 100 million to 1, you've hit the jackpot. You can publish your Nature Genetics study that finds a genetic association. So the first time genomic screening was applied to images was about 3 or 4 years ago. Some genetic variants were related to temporal lobe volume. They were interesting. At least one of them was already a target for Alzheimer's therapy in the glutamate receptor gene. They found that GRIN2B is a polymorphism of the glutamate receptor that was already a target for therapy. And you can find out what these genetic variants do. You can do a regression of each point in the brain against signals and look at the pattern of effects. This one seems to be associated with atrophy of the temporal lobe particularly. And you say, well, if you need thousands of people to find these things, do they really matter? I mean, if they're so difficult to find. Well, here's one that really matters. So 1% of the people that we know carry a genetic variant in TREM2 that doubles your rate of brain tissue loss. This was only discovered very, very recently. Priya Rajagopal and Derek Heibar, who's here today, England Journal of Medicine published this. That's a hell of a hit. It's a rare variant, or not so rare. So in ENIGMA, people take MRI scans, parcellate them, take DTI scans, measure tracts, test genetic associations, and meta-analyze the results. And they upload the meta-analyzed results here. So the first paper in Nature Genetics basically said, if you look at things in the brain that are very general, like the size of the brain, or the size of the hippocampus, where is there in the genome that can predict the size of these structures? And one of the variants that's found here, the p-value is pretty good, and there's 21,000 people in the study, so the p-value is excellent, was able to predict about 3 or 4 years of aging in the hippocampus, the memory system of the brain. So knowing your genotype there will give you a good idea as to whether your brain age, so to speak, is normal, or 3 years advanced, or 3 years behind. Now this is a forest plot, so I want to say what this is. So each of these cohorts, or groups of people who've been scanned, can vote. And so if you vote for the president, certainly in the US, the Electoral College is a way of calibrating votes from different states and picking a president. So I don't know, the analogy is probably not good. But each of these cohorts can say what is the evidence in that cohort for a certain gene or a certain feature being associated with a feature in the brain, here, the hippocampal volume. And in the end, the evidence is just outrageously strong. In individual cohorts, the evidence overlaps with zero evidence, but when aggregated, it's just unquestionable. So I'll skip that. So these studies are getting bigger and bigger. There's a collaboration with Charge. Derek Ibar, who's here, is doing this one, certainly the largest brain imaging study ever. Areas of the genome that are just extraordinarily predictive of features such as hippocampal volume. Again, just the Manhattan plot. The reason it's called that is it's like the buildings on the Manhattan skyline. It's these little chromosomes that are numbered here. And what this means is that there's an extraordinarily low p-value or good evidence for this genetic variant in being able to predict the volume of your hippocampus. So some of these are disease-rich genes, and maybe you can screen images and discover genes that cause disease. All right, so that was all a lot of neuroscience. So now we're going to do the engineering and mathematics. So that wasn't very efficient. It really took about 26,000 people's data, and this is Derek Ibar's data on ENIGMA 2, where the volumes of certain parts of the brain, you can imagine each piece of the brain has a Manhattan plot in it that tells you genetic variants that relate to or predict the volume of that part of the brain. So an image is made up of about 8 million pixels. And these are only 8 pieces. So let's take this idea a little bit further. So you can look for genes that affect the brain by screening the entire brain and running a genome-wide association with each pixel. Now, this is a hell of a computation. It'll take you a very long time to do. So you run a genome-wide scan of every voxel in the brain. The scan could be for functional image. It could be integrity of the brain on DTI. You can split this problem up so that the images are matrices. You can send them to different computers and they can chug away. But you do have to correct for the 138 or whatever billion statistical tests, because you're asking the question, does each variant predict anything in each pixel? And just by randomly coming across things, you need a p-value that is 10 to the minus 12 to find any rigorous evidence for the linkage. So how do we make this a little better? So this was done. Jason Stein, who's a postdoc now in our group, kept the most associated gene at each pixel in the brain. This is a plot of volume, and kept them and looked at the functions of these. But again, it's not a very efficient way if it takes 10 days to run a search of the genome, a search of the image database. So Neda Jahanshad in our group, and she's there at the back, she was interested in what is the power to detect a gene effect in an image. Can you calculate it? Because NIH often wants to know, well, if you scan 1,000 people or 10,000 people, are you ever going to have a realistic chance of finding the things that you care about? So this is an interesting analytical study that she published. It's very well done. That shows that your power to pick up a genomic variant effect in an image, if you search the whole image, depends on the heritability, which is the proportion of the trait variants due to genetics, sample size, obviously, something called the minor allele frequency cutoff, which is the rareness of the gene. You don't want to have really rare genes because they're not useful. And this magnificent formula here can be run, and you can tell you how many subjects you need. So you kind of need about 600, 700 subjects to have any chance of finding associations at all. If you have fewer, you should join ENIGMA and pull your data with everybody else. So there's a lot of ways that pattern recognition has been used to make sense of lots of images. So you all know about automatic labeling of images. So when you automatically label images, you try and cluster things that are coherent. So we know that genes act in the brain in a coherent way. They don't affect one pixel. And so one of the ways that these things have been recovered is to look at what is called genetic correlations in images. So let's say we think that the size of the brain and IQ are correlated. It's true, they are. And let's say we think that something about our genetic makeup causes that. There are some genes that make the brain bigger, some genes that affect our IQ. That's a more contentious statement, very contentious statement. You can look at whether that's true in twins. And so if you have a pair of twins, monozygotic identical twins, same DNA, if you can predict the other twin's IQ from this twin's brain volume, there's a cross-twin correlation. And there's a thing that genetics experts have come up with that measures the degree to which a correlation is mediated by common genetic variants. And this has found genetic links between all sorts of things we didn't know were linked. So iron content in the blood and brain diseases, there's genetic links there. Now let's take the analogy a little bit further. So let's say you're in an image here in one twin, and you look at the other twin's brain image over here. Now that's a really strange thing to do. You're looking at a pair of scans of two different people. But if you can predict anything about the other person's scan in the other location, from this twin scan in one location, you have a genetic association between them. And if you're better able to do that in people with more DNA similarity than people with less DNA similarity, you know genes are causing the link. So you could sit in a scan and say, tell me all the voxels, all the signals, all the other measures that we've done that are genetically correlated with this voxel, and we'll clump them, we'll cluster them. And you can do clustering. And so when you cluster these images, you'll get lumps. And the lumps are based on a twin design way of asking questions about similarities. And this is very efficient. And so this is work by Bing Chang, who was a postdoc with me, that was able to find genetic variants that affect PTI by clumping regions and basically clustering the genetic correlation matrix of all pairs of voxels in an image. Boring. So there's a problem with doing these things in ENIGMA. So genetic data is private. So if you have an illness that you don't want other people to know about, or you don't want your insurance company to know about, or your siblings, a study will often have you sign a consent form that says, this data will never be sent to any other institution at all, even if it's anonymized. So one of the ways that we're beginning to work with the folks at ISI, the marine engineers there, is to try and fit predictive models to data that's somewhere else in the world. They don't send us the data. You can make a prediction about a gene or a disease affecting the brain. And then they send back their local results to a central server at USC for meta-analysis. So you could imagine those genome-wide, image-wide search that have about 1 trillion results. If you have 30 centers in ENIGMA around the world that are competent to do this computation, this is a really good parallel computation. So there's no message passing. So most parallel computations involve, if you solve a PDE for a fluid flow, each flowing piece of the fluid is connected to everything else, and you pass information on gradients to all of the other processes. But this is not a synchronized computation. It's one that's done distributedly, and they're passed at the end. This is how ENIGMA works right now. But could you do it a little bit better? So coordinate machine learning, which we're also looking at, is you're mining images for signals that are very difficult to find. So could you fit a local model, a predictive model of each site, each of these cohorts in ENIGMA, but constrain the coefficients or the predictors to be the same ones across all of the different sites and just pass the model coefficients around iteratively. So this is sort of computer science way of thinking about this, where you integrate the data. Now this model passing helps because genomic data is typically private. You won't find a lot of it on the internet, but you're allowed to do certain restricted queries, not every single letter of a person's genome, but at least a certain number of them. And you can capitalize on the fact that all of this machinery is somewhere else, and you don't have to pay for it. So one of the ways that this can be implemented using machine learning is called a distributed lasso solver. So you remember earlier in the talk we talked about predicting things in the brain from very, very weak signals, so things on your genome. And one standard method is called lasso, and the mathematics are fairly straightforward. It's a least squares fit between your predictions and the truth. The term on the right there is an L1 norm. It's used to constrain the model coefficients, or the set of predictors, to be as efficient as possible. You prefer a model reality that has more compactness than one that has a lot of details. So Jieping Yi at Arizona State said, you know, this is a tricky computation to run at many, many sites in Enigma. There are certain types of machine learning problems that are separable. In other words, if you don't have all of the data, you can split it up and run the algorithm at each site, and you're no worse off than having all the data yourself. So he had proven, and it's a very useful result for Enigma, that if you can write certain machine learning problems such as this one, so that the objective function is separable, you can iterate over all of the sites in Enigma and have the gradient of the objective function, in other words, the nudge to the parameters that you give to improve the solution, and send those back to an Enigma site, this central site at USC. And each of these sites just gives you the gradient or the nudge, and you can compute on all of the world imaging data without there necessarily needing to be any transfer of data from one site to another. Each site only provides the gradient information, and the data matrices of all the genome's images don't have to be transferred across the sites at all. This is also quite good. So if you don't have a computer that can fit 26,000 images in its RAM, which is very usually the case, you're using a lot of the other infrastructure as well. So you can do better. So there's another way of doing machine learning in distributed learning called asynchronous stochastic computation. So let's say you have collaborators and it's Christmas, and you want them to edit your manuscript. Some of them will reply, because they don't celebrate Christmas. Others, you'll have no idea where they are. And that's what happens in Enigma. So there's plenty of sites out there where you've no idea where they are. Maybe they're on Safari. So the problem with having a distributed computation that requires synchronized replies from servers is that half of them are down, or you don't even know what's going on. So there are forms of machine learning that update parameters in a stochastic way. And we already said that this data is all remote. Each of the sites can pass their best estimate of the nudge for a model. When I say model, it's very general, so it could be an imaging feature that is linked with disease. It could be a gene that's linked with a brain image, or it could be a gene that's linked with a disease. So we're just starting to test the accuracy of this. So this notion that you sort of split the data, it can't be shared either because people don't want to or the consent prohibits it, but you can update a parameter server where a very small amount of data is passed. It's tremendously useful. The stochastic element allows sites to poll. You can poll sites when they're up. If a site is down, you don't need to use their data. So you can actually check whether this works because you could split a giant data set into n private test sets